Thank you very much, Arvi and Jim and others, Swaroop, for the invitation. It's great to be here. Okay, so here we have my version of the several levels of investigation that we how we can approach to study the brain. And among the basic levels, and we have already heard this from Steve Peterson's talk, are molecular processes, including neurotransmitters and receptors, and single neurons. The brain consists also of, or has many simple neuronal circuits, such as a reflex arch, and the reflex arch is something that we understand quite well. And then there are complex neuronal systems, right here, and they consist of large assemblies of neurons that together function as a whole. And I think that a lot of the cognitive neuroscience research currently is aimed at understanding the functions of these networks and how they influence behavior, how these networks change during the life of people, and how they are affected by psychiatric disease and neurological disease. And it is at this level of these large neuronal circuits that we work when we're using functional connectivity MRI. So that's what I am focusing on, as many others, of course, at this conference. Now this is a slide that I included because I did not know the exact composition of the group. It's a little bit silly slide because I'm in the room with giants of neuroimaging. But I'll go through it anyway, maybe as the introduction for the novice who's entering the room today. This slide shows the difference between task-related fMRI and resting state functional connectivity MRI, the latter often abbreviated as FC MRI. And the typical task design contains of periods of a task, in this case an example of looking at an image, and periods of rest. And then the data during the rest, in this case eyes closed, is subtracted from the data that was collected during viewing the images. And after applying a certain threshold, this map shows up and you see the visual cortex being more activated during the task than during rest. So that all makes sense. And it is these methods that have spurred scientific discovery immensely over the last 20 years. And it is these block designs, but also complex event-related designs that have been very instructional, and we've learned a great deal about it. But resting state functional connectivity MRI works differently. So then here there's no task, and the brain at rest shows large fluctuations. And if you would measure the signal from, for instance, this region here around the motor cortex, you can plot that signal, and it's plotted right here. And if you compute the correlation between that signal and every other voxel in the brain, apply a certain threshold, then you will find a map like this. And you have the somatomotor system, because the seed region was a little bit over the precentral and the postcentral gyrus. So this is basically what we are looking at with resting state functional connectivity MRI. This is an example of seed-based analysis, but as many of you know, there's also a different class of analysis. You can use independent component analysis to extract, and I think the same kind of signals. So they're also very suitable for this kind of work. But it is important to realize that resting state connectivity analysis can look at this network, like the motor network, but also other networks, without that the subject is using that network. So that is, I think, one of the big strengths. And this is another demonstration of these correlation analysis. The maps in hot colors here, from red to yellow, show the correlations with the seed region. And the seed region is here, a little black circle. And the seed region is located in the lateral parietal cortex and shows a distributed network, we've already seen this today, in the medial temporal plus complex system, and also here in the premotor areas, the frontal eye fields. So this is an example of the dorsal attention system that you see. And what I will now show you is the functionally coupled regions with the seed as the seed is slowly moved through the parietal cortex. And as anticipated from monkey anatomy, there are multiple networks, and they are quite distinct from one another. That is, areas close by in the parietal cortex are connected to functionally coupled regions spread throughout the cortex. And here's, I think, a nice example where the seed region is in the center of one of the nodes of this famous network, the default network that we're all looking at. You see, this region is highly functionally connected to it. And if the seed region moves a little bit further, right there, it's close by. And in an area further away from the seed region, a different region right here, it's more the retrosplenial cortex, seems to be correlated with that point. While when it is here in the center, you'll see the precuneus, or the posterior cingulate region, light up. So regions very close by are parts of networks with distributed different signatures. That's the point of this demonstration. So here is an example of a parcellation of the brain into 7 major networks. And this image comes from our own group, but many other groups find similar parcellations. Also mentioned this morning by Pierre. The colors are a little different than on my screen, but we see in blue the somatomotor system. Purple is the visual system right here, spanning the occipital cortex. In green, we have the dorsal attention system again. This yellowish is the frontal parietal control system, as we like to talk about it. And here in pink is, again, the default network, or the default mode network. And Fenna Krienen, together with Thomas Yeo and Eun-Young Choi, have done most of the work. And this was all published in the paper by Thomas Yeo et al. In the Journal of Neurophysiology. So Fenna is also here at the meeting. There she is. And she has her poster up. And she has her poster on clustering analysis, breaking those 7 networks that I just showed you up into subnetworks, providing insight into cortical-cortical connectivity beyond the parcellation that I just showed you. Her poster also touched upon models of hierarchical processing that have been shown in monkey cortex, in the primary sensory cortex. And she demonstrates that resting state functional connectivity, MRI, can be used to infer hierarchical structure of visual motor pathways in humans. So if you haven't had a chance yet, in one of the next breaks, we definitely check out that poster or talk to Fenna about it. Here's the outline of my talk. First, I will present evidence indicating that we're measuring actual large-scale distributed brain systems and show you some data on reliability of FC MRI. Then I will touch upon the topic of confounding effects of head motion, that were also already mentioned this morning. And after which I will go on to talk about one of our studies in which we predict advanced aging using multivariate pattern analysis. And finally, I will show you some preliminary data indicating the advantages of high spatial resolution functional connectivity MRI at 7 Tesla. So, a particularly good test case to determine whether functional connectivity MRI captures, at least in part, existing neuronal systems are cerebellar circuits. The anatomy of these circuits are well established, as illustrated here in this schematic. And the cerebral cortex projects to the cerebellum via a polysynaptic circuit that goes through the pontine nucleus right here and crosses to the contralateral cerebellum, and then goes back through deep cerebellar nuclei, through the thalamus, back to the cortex, and back to the original part where this first signal originated, forming a closed loop system whereby the input to the cerebellum also provides the feedback. And a very basic question is whether functional connectivity MRI can detect these cerebrocerebellar circuits, and whether it demonstrates the cross-lateralization expected from the anatomic organization. This work was also done by Fenna. And this is what Fenna found. She measured the intrinsic functional connectivity here in the motor cortex, And she searched across the cerebellum and found the functionally coupled regions. And here is the well-known primary representation in the cerebellum of the motor cortex, and here is the secondary representation. And when she placed the seed on the other side, she found the expected crossed pattern. Now we know from this work and by others, such as Christopher Honey and Patrick Hagman and Olaf Sporns, that functional connectivity does not equal structural connectivity. It was also in the previous talk clear that it definitely doesn't equal. But these results do demonstrate that functional connectivities in humans is capturing expected large-scale properties of distributed neuronal systems that contain polysynaptic connections. And in a study performed by Husheng Liu from our lab and others such as Ji Lu, they looked at patients with focal strokes to the pons, and the study included 11 patients, 4 with lateralized lesions to the right pons and 7 patients with lateralized lesions to the left pons. And focal strokes to the pons, such as shown here in this image, a little hard to see, but it's this little black dot. They emerge from infarcts from small penetrating branches of the basilar artery off to the side of the brainstem. And the cerebrocerebellar circuit was again the target of study because such lesions selectively disrupt the feed-forward corticopontine tract that normally crosses to the other hemisphere. The pathways to the cerebellum on the same side as the lesion, so if the lesion is here, the cerebellar region on the same side should be unaffected. So this creates a very specific prediction that contralateral, right here, but not ipsilateral connections should be disrupted. And paradoxically, the regions closest to the lesion should be unaffected. And that is exactly what they found. So lesions to the pons on the left side selectively affected the contralateral area right there. And the regions closest to the lesions were spared. And the effect was reversed in patients with right lateralized lesions. So this result reinforces that our measures are detecting selective information about specific circuits. The study I'm going to show you now was aimed to see if functional connectivity could map this topography in humans. And a group of individuals are first imaged simply performing active motor tasks. In one condition, the subject flexed their feet. In another, they moved their fingers. And in a third, they moved their tongue back and forth. And the motor regions active during that task are shown here on the left. And the inverted representation in the cerebellum is clearly visible. So then in another group of subjects, functional coupling of these motor regions at rest, so these regions to the contralateral cortex were imaged. And what emerged was the clear inverted topography in the cerebellum, now determined exclusively by functional connectivity MRI. Thus, without even using the hand, foot, and face regions, they can be mapped using resting state analysis. And here's one more view showing the primary and the secondary representations. Taken together, these results build confidence that we're able to measure the architecture of large-scale human brain systems using resting state FC MRI. So in our lab, we also performed analysis to get an idea of how stable these measures are over time. That was part of a methodological project that I took on when I joined the Buckner lab and resulted in a paper published also in the Journal of Neurophysiology in 2010. And we looked at things like, does it matter if the subject has their eyes open or closed during rest? Does it matter if you have a TR of 2.5 or 5 seconds? Does the voxel size of your acquisition matter? And all those things are in the paper. I'm now going to show you something about reliability. And here you see single subject data. So there were 6 subjects scanned twice with approximately 7 days apart. And this is scan 1 of subject 1 And this was the same subject 1 week later. And in yellow, you see the overlap across time. Now, this is the case where we analyzed 40 minutes of resting state per subject. And I'm only showing this to give you an impression if you have the perfect scan. So how reliable is it? Well, it's pretty reliable, but it's also a very lengthy scan. Now I took 5 minutes worth of data per session and plotted the same posterior cingulate seed correlation map. And it is, of course, clear that there is a lot more noise, but nevertheless are the points of overlap across time always in those canonical areas of the default network, indicating that maybe 5 minutes can already be enough to take a scan, especially if you're going to average across subjects. So to further home into that idea of what a minimal length for a resting state scan is for reliable results, we took one 12-minute run and analyzed the data with run lengths ranging from 1 minute up to 12 minutes in 1 minute increments. And here you see the average posterior cingulate correlation map for a 1 minute, 3 minute, 6 minute, and 12-minute segment.
Can you say something about the threshold? What is the statistical level? Is it because you don't specify the threshold level? Is it the
That's
same for all of them?
Oh, definitely, yeah. There's here the same threshold. It is actually a raw, it's a Z map. It's an R map converted to Z using Fisher's R2Z transformation. So it's a Z map, very close to R. And it is not, it's not a second level analysis, so there's no statistical threshold. This is a raw map. I believe it's a maybe 0.25 and higher. It was a little study, average of 6 subjects. And there are 2 main points to take from this slide. The first is that even in a 1 minute scan, the contours of the default network are already visible. And the second point is that after 6 minutes, adding more data does not buy you a lot more signal. That's what we think. And I can quantify that in a different way. And here is depicted on the y-axis the correlation strength for several networks. The solid red line is the default network. The dotted red line is the attention network. The green line is the average correlation among 3 regions among which we expected to find low correlation. So those are correlations between the visual, motor, and auditory cortex. We expected a good scan to show high positive correlation for the default network, attention network, but a correlation close to 0 for that reference network. So that is the network among visual, motor, and auditory cortices. And what you see here again is that the default network signal was right there from the beginning. It remained somewhat stable. And that the green line is also around 0. You do see that the error bars decrease when the run length increases. And the attention system, you see that the correlation seemed to become a little stronger. And from here on, it is roughly stable.
Can you say where the error bars, is this across subjects?
Yes.
So there were 6 subjects?
Yes.
And those are standard errors?
Standard errors of the mean.
Thanks.
Yeah. And so the standard errors bars, you see these decrease. And another way to get a handle on the noise properties of the data, so the signal seems to be there for a major network right from the beginning, but there's something with noise about these, if you see these error bars. And one way we also looked at this was to measure the connectivity values with the posterior cingulate seed region in regions where we would not expect high functional correlations. And that is depicted here in this yellow map. And from that mask, we took all the average correlation strength, and you see that decreased proportional to the square root of the sampling time. So, and that is what you would expect from a measure of noise. So while the confounding effect of motion on MR imaging has been known for decades, almost no one in recent papers was talking about the effects of the possible confounding effect of head motion. And what I would often read in papers, and also when I would review a paper, people would say, well, we excluded people with excessive motion, period. They would not say how many people were excluded, for instance, in the patient group, or how many in the control group. They would not specify what the definition of excessive motion was. So I really thought that there was a lack of attention to that problem. Especially if you think about it, that if you would say, well, everybody with motion more than 3 or 4 millimeters, we'll exclude them. It doesn't mean that under that threshold, that if you have two groups, that the motion is the same. So that's what we went after. And earlier this year, several papers came out and in neuroimage that put this issue of head motion in the spotlight of people working in this field. And Jonathan Power from Steve Peterson's group demonstrated the issue and also came up with a solution by suggesting to remove certain volumes where the motion occurred. And there was also a study by Ted Satterthwaite where he demonstrated why motion is particularly problematic if you study children from different age groups. And I will now show you some data of our own paper that was entitled The Influence of Head Motion on Intrinsic Functional Connectivity, MRI. So in this paper, we measured head motion in 3D space during the 2 resting state runs. And this illustrates head motion in the scanner, and I'm showing you the estimates of 3 subjects. The blue line is a subject that moved very little. The red line is a medium subject, and in green is a subject that moved most. And these are all young, healthy, college-aged, very compliant study participants, because you can see that none of them really moved. There was no gross movement. There was never more than 1.5 or 2 millimeters motion.
Excuse me, is this here?
Oh yeah.
Is this an actual measure of head motion, or is it an indirect measure, looking at the images or some other?
These are the measures taken from the motion correction parameters from the rigid body motion correction. Yeah.
It's not motion.
It's an indirect motion. Yeah. That's a great point.
It could be functional activity.
It could be. Yes, although we do know, I mean, in general, it maps pretty nicely on actual motion. I mean, if you just watch the time course, done that many times, you play a little movie of the time course, if you see a jump like that, you'll see it in your image, and it is a little shift, you know. But it's a great discussion, and it was also already mentioned this morning. It would be very nice to have something like a little measurement thing on the head that you could maybe optically measure. So those are definitely things to think about and improvements for the future. But okay, so this data. We estimated head motion in over 1,000 subjects from a sample of healthy college-age adults collected in our lab. And on the left side, you see the distribution of head motion across the sample. And there are 2 things to note here. The first is that males move more than females. You see that here by the shift of the open bars. They're shifted a little bit to the right, and the females, the black bars, are shifted to the left. And the second thing is that the bulk of the subjects, of the healthy subjects, are very compliant subjects that lie very still. On the x-axis, you see mean motion, and now it is relative motion, the average relative motion over 2 runs measured from one scan to the next. So now, yeah, the total group was then divided into 10 bins, ranging from decile 1, containing the 10% subject who moved least, to decile 10, containing the 10% who moved most. And then we computed the posterior cingulate correlation map of the subject from decile 5 and from the group that was in decile 6. Those 2 maps that I don't show here look to the eye very, very similar. When we subtracted, when we did a subtraction between those 2 groups, like you see right here, the default network appeared. And So this and this effect, you could say, could in a different context easily be confused for real neuronal effects. So you could go and interpret this as there is a difference in the functional network properties. And so we believe that this is a direct effect of motion. So these are the results shown in another way. And on the left here, you see the average default network connectivity strength for all the subjects. And you see that the average default network connectivity decreases when head motion increases. And you see the same effect for the frontal parietal network. So let me be clear in one thing, that these effects of head motion are evident after all the standard pre-processing. So of course there's the rigid body motion correction that was performed, but we also regress out those parameters plus their first temporal derivative, etc., as many people do. So these are residual effects of motion. Yeah.
Correct me if I'm right, but it's saying that motion going against you, meaning that the correlation decreases, so it cannot generate effect, it's simply you losing effects. This is what you're showing?
Yes.
Okay.
Yeah. So the functional connectivity of that network decreases. But now it turned out that head motion did not have the same effect on every measure that we looked at. Because increasing motion here was associated with a slight increase in functional coupling between the left and right motor cortex, and also showed an increase in local functional coupling. And local functional coupling was actually a measure of local degree connectivity. What I should say about one slide back, that here a nonlinear fit did not give us a better, more significant fit than the linear fit. Although here you see nonlinear fits that were slightly explaining more variance. So what we see is that motion may have systematically different effects on functional connectivity depending on the network studied. Interim conclusions: first, head motion has significant effects, and second, that most variance in FC MRI metrics is not related to motion. And you can see that clearly here, that people, a whole group of people that moved similarly, had a very wide range of functional connectivity values. So there is a lot more than motion, of course, in these signals. However, since the effects of interest in FC MRI are often between groups of subjects where differences in motion are expected, such as children versus adults, young versus old, and patients versus controls, it is very important to consider these effects of head motion in your study. And in addition, we should look at improvements of the effectiveness of current regression techniques. And I know that there are some papers under review right now doing exactly that. And we should definitely consider methods like the scrubbing that was already talked about this morning, that is nicely described in the paper by Jonathan Power. So now I'd like to switch gears a little bit and show an example where we try to predict advanced aging using multivariate pattern analysis. From several previous studies, we already know that functional brain networks are affected by advanced aging. So here you see two examples, one from Damoiseau and colleagues, and one from Jessica Andrews-Hanna, who at the time was a graduate student in Randy's lab. And both these papers indicate that on average, older adults show a decrease in functional connectivity strength of the default network. And you see it here in an image form. And the aim of the present project was to explore whether functional connectivity patterns have any predictive value for individual participants. For that, I selected all the older adults, all the people over 65, could also call them mature adults, and I selected a younger control group, and they were matched not only based on gender, but also based on mean motion. So these two groups showed similar motion. And the multivariate pattern analysis consisted of 3 steps. And now I realize that in the room here, many people will know a lot more about these methods than me, because I'm just getting started into these topics. And it was, I should also acknowledge, Mert Sabunju, who did all the actual analysis. So there are parts of black box in here that are black boxes to me and probably not to many people here in the audience. But the steps that we took. First, I defined a total of 450 spherical ROIs, evenly distributed across the cortex and the cerebellum. This is funny because this is somewhat of a grid way of doing things. Then I computed the full correlation matrix of the n by n matrix, so the 450 by 450 matrix. And this procedure is similar to the Dozenbach paper that was published in Science in 2010. Then in the third steps, these 100,000 features that you have in this upper area were analyzed by a linear support vector machine classifier with a leave 10% out cross-validation in order to discriminate young versus old. How this SVM machine learning algorithm exactly works is, again, some things are somewhat black box to me, but let me just try to explain it a little bit in simple terms, that the support vector machine algorithm tries to learn a decision rule to classify data into groups, in this case, young versus old. The linear SVM method employs a plane or a hyperplane as a decision boundary to optimally separate two classes. And in this toy example, you see a two-dimensional space where the data is trying to, or the classifier is trying to separate it in two classes by this decision line, this red line. That's the decision boundary. The SVM score is the distance from where an individual subject lies in the multidimensional space, here 2 dimensions, and the decision line. Okay, and in this toy example, you see that all the participants, if these are participants, for example, are classified correctly, except this one example that was misclassified. So here you could say that the algorithm had almost 100% accuracy, but was just short of 100%. And here are the results of the analysis. On the x-axis, here you see chronological age, and on the y-axis you see the SVM score. And these results indicate that the SVM accuracy was almost 95%, so that only a few subjects were misclassified. So these were misclassified and those. And both sensitivity and specificity were above 93%. A second point was that within the older group, the SVM scores were significantly negatively correlated with chronological age, right here, shown by this regression line. That was a correlation of 0.41. And that was kind of a bonus finding because the SVM classifier was not trained to find that. You could also train a classifier to do that, but this one was only trained to dichotomize the two groups. But then it turned out that this worked out also for chronological age. That was a nice validation, I think. And in the light of those recent reports on the effects of head motion, we wanted to check if motion had any effect, despite the effect that the groups were matched on motion. So here you see the SVM score, and here you see motion, and they were not related. And in a subgroup of older adults, we also had a DTI scan. And here I computed the mean fractional anisotropy in a mask of the deep white matter. And that measure positively correlated with the SVM score. So again, some interim conclusions. From individual subjects' resting state scans, we could reliably predict if that subject was young or old. The SVM score showed a correlation with white matter integrity, which might indicate that these functional results seem to be picking up actual structural brain changes in some of these older adults. And finally, that resting state functional connectivity measures may prove useful in predicting brain pathology in patient groups, because the data I just showed you was really meant as a proof of principle, because predicting someone's age is not as interesting as being able to predict from a resting state scan whether a person might have pathology. So those are, I think, one of the promises for future research that we definitely want to try out. So I came to the final part of my talk, which is last project that I started working on. So as many of you know, we have this so-called Connectome scanner at the Martino Center here, and also the 7 Tesla MRI scanner. And the Connectome scanner is currently being optimized to do diffusion imaging to get at the structural connectome of the brain. But all the data I show you today is from the 7 Tesla MRI scanner. From this image, you can already appreciate the high spatial resolution, because I'd like to point out that although you see some activation from a finger tapping test, the gray underlay is the first bold image of the run. So this is the raw bold image, the first bold image. Here is now an example. All the data I'll show you is single subject data in this person's native space. And here's an example of one subject showing in hot colors the activation during right hand finger tapping. The contralateral motor cortex here is activated, and the ipsilateral cerebellar region, and we've already seen before in images. There's a question. Oh, 5 minutes. Good. Thank you. I think what you can see here is that, there is an amazing spatial resolution. You can recognize the inverted omega of the motor strip. And I would say, it's an amazing improvement over standard images. This is an image of a publication of 2006 of a finger tapping test. This was a group of subjects. This is an individual subject. But I think this is, pretty cool data and very promising for the future. But the main question for me was whether we can see the major functional networks during the resting state. So without asking the subjects to perform a task that rely on these networks. And here is the detailed functional connectivity map that you can get. And this is an independent component analysis on the data of 2 sessions, also 1 week apart. And this is the map that I identified as the motor strip. And then I used the region at the crosshair right here as a seed region for session 1 and for session 2. And you see a pretty strong overlap across time. And again, this is an image of our paper of 2010 showing the motor system. And what I really like to point out is that we've always talked about this system as the motor system or the somatomotor system. And this is the system where Bharat Biswal in 1995 started doing the resting state functional connectivity analysis on. And we were starting to see in this data that only the precentral gyrus was showing up in this independent component map. And in another map that I'm now not showing here, we saw the precentral or the postcentral gyrus. So possibly pointing to distinctions between or separations or subnetworks within this large blob that you see there. And in addition, major systems such as the default network and the attention system were each represented by component maps that clearly followed detailed individual gyral morphology. And so these results provide evidence that high-resolution FC MRI can be used to estimate detailed maps within individual subjects. Lastly, I'd like to show one more result of a single subject. The data was all pre-processed using our standard pre-processing pipeline, with the only difference that the data was not normalized to standard space, so no atlas space. And I will show you now a movie where you see me move the cursor through one slice using AFNI's InstaCore. So instantaneously, correlations will be computed with the location where the mouse is. Oh yeah, there, I stopped it. So now I'm moving through the deep white matter, and when I hit the cortex, you see the usual suspects pop up. So here I'm going to search for the motor strip again, precentral. There it is, precentral. And when I move back, you see the dorsal attention system right here. I move on to the default network, and finally to the visual system. So I've shown you that there are a number of pitfalls to keep in consideration when using resting state FC MRI for the study of large-scale brain systems, and that these pitfalls definitely warrant more methodological investigations. And I hope that I've also been able to convince you of some promising new avenues, such as applying MVPA to aging and disease, and also going to higher field magnets. So quickly, my acknowledgement is first of all to Randy Buckner, of course, my current supervisor and mentor in all these projects. And I'd also like to thank all the people that have been helpful in the several different projects. Thank you very much for your attention.
Koene,
Yes.
on your support vector machine analysis, the multivoxel pattern analysis, what were the brain regions or the brain connections? Do you have a slide that shows the ones that were driving those age-related differences? Projected onto the brain?
Oh, no. So I don't have that. I do have another image. Now, I did keep this one, so it's a little misleading because this is not based on any MVPA analysis. This is simply the posterior cingulate seed map showing which voxels show a significant decrease with age. And this is the number of subjects, older subjects, that showed a significant decrease with age. So if you just want to know where is that happening, because we know it's happening somewhere, where is it happening? It must be in these areas. But it's not a finished analysis because this is, of course, just with the posterior cingulate seed map. So I don't have the definite answer to that. That's a good point to continue.
I liked your cerebellum example, showing nicely the structural layout of these functional connectivities, but I couldn't help but notice that the thalamus was missing. And I thought that there may be two reasons for that. One is the signal to noise is not high enough, and the other one is maybe the thalamus is kind of doing too many things to be correlating with these two other regions. And I was thinking that then it's a small question for kind of hub people. Is it possible to actually detect hubs with these techniques if by definition hubs are doing too many things at the same time with too many networks, and whether you can apply this static correlation analysis to detect them, or whether you have to go to more like dynamic type of analysis?
Yeah, well, it's a great question. I don't know exactly what the answer is, but maybe Fenna, you can give us some.
Yeah, this I don't remember either. Either this was masked out, or it's the wrong slice to see the appropriate part of the thalamus. You can certainly see correlations in the thalamus, but I think functional connectivity really suffers when you have a very small nucleus to disambiguate different networks. So we've done some work trying to parcellate the thalamus. But it's certainly there. The correlations are there, but it's hard to see. The correct laterality is there too, but it's probably masked out in this image.
Yeah, I was very happy about your result, like of motion between men and women for some reason. Is that in your recent neuroimage paper?
Yeah.
Yeah, okay. Because we had observed that in our lab with, I think, a dataset which comes from Cambridge, like the one which is a 1,000 functional connectome.
Oh, you've seen it in that data?
Yeah,
That's
yeah,
not this data, that's different
okay.
data.
Because we have analyzed it in the lab, and I was looking at sex differences, and after regressing out the effect of motion, everything disappeared. So I noticed that, except that we tried to replicate it in about 10 other sites from the 1,000 functional connectome, and we couldn't. So I'm sort of like curious about how do you recruit your subjects exactly? Something.
They're all undergraduates, and I don't know. It's a point we did not dive into.
Okay.
And one of the things is, by the way, in my comparison, where I show a significant difference between high movers and low movers, those two groups are equal, men and women. So
Okay.
it's not fully explained by sex or something.
Okay.
But
well, an hypothesis I had, and we haven't tested that, is that, I mean, one of the big difference between men and women is that women are a bit shorter on average. And maybe that has an impact on the size of their brain, even though I've never really tried that. And I mean, the motion for a given rotation, a big brain will move more. So it may be related to brain size, if anything.
that's an interesting, hypothesis. It could be right, yes.
Isn't there also the possibility that there's a difference between people who fidget a lot and people who don't fidget?
Yeah.
And so it's not just a motion artifact, it's actually an individual difference in the functional connectivity in the default system. And the fact that it was only in the default system and not in the motor system
Yeah.
or the other one, I would think that if it's just an artifact of motion, you should see it across all systems rather than just in one functionally specified system.
We definitely discussed that, and I don't think we can exclude that possibility. Although I would think that, these results that large-scale systems show a decrease and local connectivity, and not only our paper shows that, but you see that in other papers, the local connectivity increases in all sorts of areas in the brain, seems to be speaking for the idea that it is just a motion and it is bad data. One second point, that's another slide I did not put up yet, is that the mean motion was strongly negatively correlated to signal to noise. So temporal signal to noise over the scan was strongly related to mean motion. So the more you move, the crappier your data is. So that gave me an idea of, look, this is signal quality and not, it's not that the brains of these people work different. Although you cannot really exclude it. So it is a possibility, although I would think it's a remote possibility.
So,
Any more? Yeah, oh, Gagan.
Dijk. Oh. So given this slide and then your slide about optimization, I'm just curious about your thoughts of whether or not that 5 minutes is still sufficient in terms of the amount of data you need.
Right.
So if one of the solutions is frame censoring, then, and you only collect 5 minutes, and 5 minutes is the lower bound, then that's a problem. And the other aspect is if you've highlighted some systems that have greater temporal SNR, like the default and the attention system that was in that slide, you still don't know whether 5 minutes will be enough to capture the stability of connections in other regions. That might be on the tail end of this graph.
I'm glad you bring that up because I actually forgot to mention that. It's an excellent point because we say, well, 5 or 6 minutes, that is the minimum you should take, although you could discuss maybe that. And that is in young, healthy subjects that are very compliant. But so, of course, more data is always better. And that's the same thing as Steve Peterson was saying. If you can collect 10, you better collect 10. And if you have time for 15 or 20 minutes, it's always better. Yes, so always more.
Those estimates can also be affected greatly by smoothing. So how large is your seed region? Is it a single voxel or is it a 1 centimeter sphere? And how much did you smooth your data before you calculated the resting state correlations?
Yeah, definitely. So there's the spheres, I believe 6 millimeter spheres, 6 millimeter radius, and smoothing is also 6 millimeters.
So if you had a 2 millimeter radius and a 2 millimeters, 3 millimeter smoothing, you would probably need a lot more time to get a stable estimate, a lot more scanning time.
Yeah,
Isn't that right?
that could be. Smoothing improves your signal-to-noise ratio.
Greatly.
Greatly, yes,
Yeah.
yeah. So that, we kept that, of course, constant. Goes without saying, if you do the comparison. But yeah, we took standards, very, very standard smoothing. But it's another thing to keep in mind, yeah.
So I don't remember from your paper, but one of the possible explanations for this is your pre-processing pipeline is coupling your motion back into your data. And as the motion goes up, you are contaminating your data by the motion corrections you're using, and therefore your signal to noise goes down.
Yeah, those are definitely possibilities.
Right. The major, in our hands, looking at some of these things, the major culprit there is motion parameter regression. And we almost never find a case where it's a good thing to do.
Really?
Yep. And there's some old literature from Roger Woods showing that even after you've motion corrected your data, if you go ahead and do a principal component analysis of your time series, the first component is motion.
Yeah.
After you've corrected for it. It's still there.
Yeah, I would definitely agree that the techniques will not remove it all, you know, because that's what this is also showing. The second idea is, can it introduce additional noise?
No question it can do that. It's just whether that's the case.
Does it do that?
There's a literature showing.
Yeah. Does that do that, and does it explain these results? I haven't looked at it. It's a great point, actually, because I just take out that one step and redo it. But
it really doesn't explain most of the motion art. Most of the punctate, small motion, DVARs kind of artifacts are not explained by things getting aliased back in.
Do you do motion parameter regression?
We've done it with and without. We've done it temporally filtering before and after. We've done it a million ways.
Okay.
You know, it's there. It's not terribly enhanced by some of the things that people claim are clearly enhancing it. It's the pits.
oh, one more question, yeah.
For your resting state fMRI study of aging, you showed some correlations with white matter changes and so on. Did you look at cortical atrophy? Because at the end you talked about low resolution
No.
fMRI. So what's the role of atrophy in these resting state networks?
Yeah, that's also a great point. Here, there's not at all any correction for possible atrophy. Those are topics that are of active study, actually. There are these models that also say that functional changes come first, and then come structural changes. That could go a little bit against that, but that's just within an individual subject over time. Across subjects, I mean, there's definitely atrophy within the older group. I've not corrected for it in this analysis. I've done it before in little different analysis, and usually if you take something like average brain size or also one measure of atrophy, it doesn't really change this result. But I see many papers and people are not at all thinking about correcting. And you should actually probably in the future think about correcting at the voxel level, because why not? You have a structural scan. So I think those are great points to keep in mind for future studies. Yeah.
Where are these white matter changes happening? Are they just adjacent to your ROI?
This is a mask of the white matter, so all white matter of the brain. And it's all major white matter bundles coming from a map, from a mask, from an atlas. It's actually the Oxford atlas that you get with FSL.